So today I'll be talking about our work in molecular models of plant hormone signaling. So first I'll give an overview of our research group's focus, and then I'll talk about the critical role of blue waters in our research. And then I'll give two examples of systems we're working on, plant signaling systems, and finally I'll talk about a computational method we've developed in order to help us study plants. So first off, so why study plants? Why focus on plants? One reason is drought. So drought is a continuing problem in the United States. This is a map of drought across the U.S. as of June 7th of this year. As you can see, there's severe drought right now in Southern California, as there has been for a long time. Below that is shown the drought coverage by year of the United States. And as an example of the impact on crop production in the U.S., the 2012 drought covered 80% of U.S. farmland. And led to severe drops in the projected productivity of crops in the U.S. And then also, so this is a plot of the yields of 3 staple crops worldwide. So the solid lines show trends from the previous 50 years, extrapolated forward in time, and the dashed lines show the projected needed yield to feed the world population, the growing world population. So this clearly shows that in order to meet these demands, we will need to produce better crops that can produce more food, and then also for the example before, we'll need to be able to create crops that are better able to resist harsh climates. So the best way to do this, I think at least, is to understand in great detail plant biology. So in order to direct genetic engineering attempts and production or design of agrochemicals that will be able to help plants be resistant to harsh climates and also produce more food. And then also, plants are interesting in and of themselves. So if you can think of a cell as a central processing unit able to take in input signals, either chemical or physical, and produce an output, then kinases would be the transistors of the cell. So kinases are proteins which are able to catalyze addition of phosphate groups to other proteins, and are also controlled, turned on and off by their own phosphorylation state. And so to give you an idea of how plants are interesting, Arabidopsis thaliana, which is a model organism that plant biologists use, is considered a weed, has about twice the number of kinases that we do, at least known kinases. So this hints at the complexity of signaling in plants. And is an interesting problem to look at then, and hasn't actually been looked at very closely, at least at the level of detail that we plan on looking at it at. So that level is the angstrom, femtosecond length and time scales, and of course, angstrom to nanometer, femtosecond to microsecond time scales. So we're zooming in on these cellular processes and looking at the individual components and looking at their dynamics at the atom by atom level with simulations. And so understanding how each of these components of cellular signaling networks work will help us then to direct genetic engineering also, like I said before. So in order to simulate or to look at proteins in this level of detail, we use molecular dynamic simulations. to look at the structural dynamics of these proteins. So these structural changes take place over very long times computationally. So we need a very large resource, and Blue Waters provides that. We're able to get a lot more simulation time using Blue Waters than other computers. And so as an example, I've shown a benchmark for, on the left is a system of the typical size that we look at, 40,000 atoms, and we're able to get 37 nanoseconds per day, which is very good. We then are able to exploit the fact that there are so many GPU nodes on Blue Waters and run independent simulations side by side and get large amounts of data very rapidly. So we're able to look at these proteins pretty quickly. In order to exploit this sort of parallel strategy of simulations rather than doing long single simulations, we use Markov state models, which take the protein dynamics that we get from simulations and cluster similar structures into states, and then we look at the transitions between those states and model the dynamics using a Markov chain. And so this allows us to use sampling strategies where we are able to adaptively choose where we seed new simulations. So this, we can't just take the simulations as they are because we're biasing the sampling. But using Markov state models allows us to get back an approximate free energy landscape of protein. So as an example, just to show you what this adaptive sampling looks like, this is just a two-dimensional potential energy surface that we're sampling. So on the left, is just a single long trajectory. So we're trying to sample this low energy region in blue, and the long trajectory doesn't leave the bottom low energy well. Whereas on the right, so if we run simulations in rounds and cluster the structures together and then seed new simulations depending on some criteria that we can choose, that allows us to explore the whole landscape. And so in this case, if we know that we want to travel in the positive y direction, we can choose to start new simulations from clusters that maximize the y component. And so then just a quick example of how we can then get free energy landscapes from that. So if you imagine that this is a histogram of simulations that you've gotten from adaptive sampling put onto a single reaction coordinate x. So this doesn't really represent an accurate frequency distribution of our simulations. So we can cluster that, say in this case 2 clusters, and then count the transitions between the clusters, and then within the cluster in a single time step or period of time. And then we can calculate stationary probabilities of being in each state, which is shown by pi, and then reweight the frequencies using those state probabilities, and then use that for free energy calculations. So those are the methods we use. One of the systems that we're working on is called the Brasson steroid signaling pathway. So this pathway controls growth and development of plants. Brasson steroid is a hormone, steroid hormone, which when it's present, it can bind to membrane receptors, one called BRY1, which is shown there. and it promotes association with another receptor called BAC1, and then that leads to downstream signaling and growth of the plant. And when there's no Bresson steroid present, or there's something wrong with the network, there's no growth signaling, and you get a dwarf phenotype, as shown on the right, the bottom right. So we simulated. We simulated these the kinase domains. So I forgot to mention both of these receptors have kinases in the inside the cell, the part of the receptor. So we simulated both of these kinase domains of these receptors in order to try to find out how they activate and potentially how the activation is regulated. Just to give you an idea of what we were looking for, so what does an active kinase look like? One that's able to catalyze phosphorylation. ATP has to be bound. You need a source of phosphate. And there's an interaction here between a lysine and a glutamate, which is conserved and important for activity. And there's also a helix, an alpha helix called the alpha C helix, which has to be positioned inward towards the center of the protein, and it has to be folded for activation. So those are the things we were looking for. We simulated these kinase domains. I should also mention that we simulated them in their fully phosphorylated forms. So if phosphorylation was sufficient for activation, we should see a very stable active state or active structure in our simulations. So this is a free energy plot made the way I talked about before of the BAC1 kinase domain. In the left-hand corner, the red oval right there, that represents the active-like state where all those criteria are met for activation. But, and I should mention on the x-axis is the RMSD of that helix from a folded helix, and on the y-axis is the distance between those two, the lysine and glutamate that needs to be formed for activation. So we see a high degree of inactive kinase in these simulations, which is a little bit, would be surprising if you're expecting phosphorylation to be sufficient for activation. So for the BRY1 kinase domain, we saw a very similar thing. High degrees of inactivity, maybe more than BAC1. So we have collaborators in plant biology at the University of Illinois, and they took the alpha C helix from BRY1, and they took that as a peptide and did circular dichroism experiments. And they found that it is in fact disordered, at least when you take it out of the protein as a peptide. Very similar, in a similar way to another kinase in humans, which called the EGFR kinase, which also has a very disordered helix. So the question for us now is whether this disorder of the alpha C helix in BRY1 and BAC1 is a regulatory mechanism. In EGFR kinases that I just mentioned, when they two of them associate with one another, that promotes folding of the alpha C helix and promotes an active protein. So our question is whether there's a similar mechanism in BRY1 and BAC1. So we'll be looking at that in the future. And then also we just wanted to see how common this unfolding of this helix was throughout the known kinases in plants. So this is a phylogenetic tree made by another group, Zulawski et al. And we mapped onto that predicted alpha C helix disorder, and it looks like there's some disorder throughout the kinase well, throughout many different families of kinases in plants. So who knows, maybe it could be a regulatory mechanism. Okay, so we're trying to create a whole model of this signaling. And so we also looked at signaling of abscisic acid signaling pathway in plants. So it's another hormone which controls water loss in plants. When it's present, it can bind to its receptor, and that makes it bind to another protein and leads to closing of pores, which control water loss in plants. When it's not present, the pores remain open and water can be lost from the plant. So this is clearly important for drought resistance. So on the right is just showing there's a very minor conformational change in the protein with binding of this hormone. So what we wanted to do is look at how the binding process happens and why binding of ABA, this hormone, activates the receptor. So we performed simulations of the binding process, and that's a free energy plot similar to before, shown on the top left. And we found that it interacts with the serine on the loop shown in the picture in the right top hand corner, and then before it jumps into the binding site and interacts with the lysine in there. But it's still unclear how this actually activates the protein. So that's something that we're working on in the future. And finally, so a computational method we've developed. So the basis for it is the idea of evolutionary coupling in proteins. So evolutionary couplings are correlations in residues, so basically appearance of residues throughout evolution. So it arises because of if you have a functional interaction between residues, and one of them mutates and disrupts that interaction, there's then a selection pressure towards a double mutant, or can be a selection pressure towards a double mutant, to then rescue that interaction and rescue the function of the protein. So this can arise either through direct interactions between residues or mediated through intermediate residues. So the idea of evolutionary couplings is then to infer which interactions are due to direct interactions, to give us an idea of the contacts that need to be formed between residues for an active protein. So they borrowed the Potts model from statistical physics, and you solve an inverse problem, and you get back coupling terms which basically tell you which residues are likely directly interacting and giving rise to these correlations. So the idea for our method is then to vary. So if these contacts need to be formed for an active protein, our idea was just to see if varying, varying the distance between these coupled residues should then take us from active to inactive proteins. So it's been used before in just structure prediction and prediction of ensembles of structures, but our idea was to incorporate it into actual dynamics. So we did a toy model, found that using this is very similar to what I showed before, using directional information in adaptive sampling just allows you to transverse the energy landscape much faster. And then we also used a Markov state model built from previous simulation data of a GPCR. So we simulated on the master equation that we got from that model. And we found that starting from the active state, using this evolutionary coupling information, we were able to reach the active state much faster using our directed method than either just long simulations or undirected adaptive sampling. And finally, we also tried it out on molecular dynamic simulations of association of two bacterial proteins. And we were able to predict okay, 6.4 angstrom from the crystal structure, so not all that great, but there's a very flexible domain in there. So we are also able to predict binding pathways in metastable bound states. Thanks to everyone in my group who did a lot of the work also that I'm talking about, and thanks to our collaborators in plant science or plant biology at University of Illinois, and thanks to Blue Waters, and thanks for listening.